In this video, you will learn about the sarcomere, which is the structural and functional unit of skeletal and cardiac muscle fibers. The sarcomere is the repeating segment within a myofibril that gives the striated muscle tissues, skeletal and cardiac muscle, a banded appearance. This unit is also directly responsible for the contractility of these tissues. At the core of the sarcomere are the myofilaments, the fibrous proteins that slide past one another to produce contraction. There are two main types, thick filaments and thin filaments. Thick filaments are found in the center of the sarcomere. They are made primarily of myosin, a motor protein with ATPase activity. Myosin converts the chemical energy stored in ATP into the kinetic energy of power strokes, the pulling force that drives contraction. Thin filaments are anchored at the ends of the sarcomere. They contain three important proteins: actin, which provides binding sites for myosin heads; troponin, which serves as the calcium sensor; and tropomyosin, which blocks the myosin binding sites on actin when the muscle is relaxed. When calcium is released inside the muscle fiber, it binds to troponin, which moves tropomyosin aside. This exposes the binding sites on actin, allowing myosin to form cross bridges and initiate contraction. The boundaries of each sarcomere are marked by Z lines, which anchor the thin filaments in place. If we look at the banding pattern, the A band is the darker region that spans the entire length of the thick filaments. At its center is the M line, where proteins hold the thick filaments together. The I band is the lighter region that contains only thin filaments with no overlap of thick filaments. This gives the sarcomere its characteristic striped appearance under the microscope. So, in summary, the sarcomere is a beautifully organized structure. Z lines mark its ends. Thick and thin filaments interact in the middle, and proteins like actin, myosin, troponin, and tropomyosin regulate contraction. The sliding of these filaments past each other is what shortens the sarcomere, and that shortening is the fundamental mechanism of muscle contraction.